Hello everybody, Antonio here, and for today you're going to learn meiosis with the help of a microslide viewer by the National Teaching Aids Incorporated. The following slides that you're going to see would have a magnification of 750 times. This means that the microscope was set at the power when the photograph was taken. So now let's begin. The first slide that you see is a primary oocyte. Each of the original 4 chromosomes has split. Each form a double-strand chromosome called a dyad. A dyad is made up of 2 chromatids attached like Siamese twins. The homologous dyads have paired. Therefore, we have 2 clumps called tetrads, labeled T1 and T2. So how many chromatids should each tetrad have? You could check your hypothesis with the next slide. Although the sperm has already entered, it is not present in this slide. So now let's go to the next slide. Still in this slide, you could see a primary oocyte. The dyads have already separated. We can see that each tetrad, labeled T1 and T2, has 4 chromatids. The twin chromatids, labeled C, may still be attached. In one tetrad, labeled T1, 2 chromatids overlap. This could be proved with fine adjustment focusing on the actual slide. Now notice the sperm, which is labeled S. In order to be a normal egg, ready to complete fertilization, it must be left with only 2 chromatids, 1 from each tetrad. How is this done? Now let's go to the next slide. Again, this slide shows you a primary oocyte, wherein tetrads would now be on a spindle. So this is an end view of the tetrads T1 and T2 on a spindle. It is clear from the positions that one dyad from each tetrad is going to leave the oocyte. How many chromatids will remain in the oocyte? So notice sperm, which is labeled S, and the fertilization membrane, labeled FM. Now let's go to the next slide. So this is a primary oocyte, labeled O, with having its first polar body forming. So when an oocyte divides, it divides unequally, like budding. The small cell about to form will be called the first polar body, which is labeled as PB1. This is called reduction division. We started with 4 dyads in the oocyte. Each new cell will have only 2 dyads. Do you realize that in each pair of dyads, the paternal chromatids are separating from the maternal chromatids? The oocyte will be left with only 1 from each pair of dyads. Now let's go to the next slide. At this slide, you could see a secondary oocyte, labeled as O, and chromosomes are now being formed. This oocyte has 4 chromosomes. The cell is ready to divide again and form a second bud-like polar body. The twins have clearly broken their connections, and each is now called a chromosome, which is labeled C. 2 chromosomes are moving out of the cell toward the future polar body. This slide shows the sperm, which is labeled S. The sperm remains inactive until the oocyte becomes an egg, ready to complete fertilization. Now let's go to the next slide. This slide shows you a mature egg. where 2 chromosomes have gone into the developing second polar body, which is labeled PB2. The mature egg, which is labeled E, now has 2 chromosomes, one from each of the original homologous pairs. Notice the first polar body, labeled PB1. How many chromatids should it contain? Polar bodies rarely divide. They die and disintegrate harmlessly. Now let's go to the next slide. This slide shows you a pronucleus stage. 2 pronuclei, or labeled P, have formed, one from the sperm and one from the egg, labeled E. The second polar body, labeled PB2, is visible with the 2 chromosomes. Second polar bodies also disintegrate. The first polar body is not visible in this slide. How many chromosomes are in each pronucleus? Now let's go to the last slide. So as you can see here, this slide is a zygote ready for first cleavage. In this zygote, which is labeled Z, the fusion of 2 nuclei is skipped. Instead, each pronucleus forms its 2 chromosomes, and the chromosomes line up ready for the first cleavage. The zygote starts with a diploid number of 4. It will divide and form thousands of cells in the new adult. Each new cell will have the diploid number of 4. The process of mitosis regulates these cell divisions and makes more certain that each new cell receives 4 chromosomes. So there you have it. You've learned meiosis using a micro slide viewer by the National Teaching Aids. Thank you for watching and listening. Till then, bye.